So good afternoon, everyone. I'm here on behalf of our head, who is not around. So he requested me to come and introduce Ranjith, and it's a real pleasure. Ranjith has been my colleague for many years, and I'm going to tell you very quickly about his background. So he has done his BSc, MSc, and PhD in physics. And this will show in his presentation and in his research because he's one of our faculty members who combines biology and physics very effectively. Um, he was a postdoctoral fellow in the US at the University of Illinois at Chicago. He was also at the Institute Curie in Paris. And he works on chromatin structure, which is how DNA winds around these proteins which look like beads, and how this is going to help the cell to sort of regulate its own functioning. So Ranjith is a really terrific scientist. He's got many awards. He's won the National Bioscience Award of DBT India in 2017 and 2018. He's won the Innovative Young Scientist Awards as well, and many such awards. So he's a really good scientist. He's also a very good teacher. He's won the Excellence in Teaching Award in IIT Bombay. And I can testify for both of these things because the first time that in our department we actually heard Ranjith speak was when he interviewed for our faculty position many, many years ago. And if I'm not mistaken, it was on Skype. And it was a horrible connection. You could not hear anything that he was saying. It was very, all crackly and terrible. And he did a great job, and we offered him the job. So I am sure that we are in for a treat today because I have myself seen Ranjith do a terrific job even under the most difficult circumstances and convince people about his research. So Ranjith, looking forward to your talk very much.
Thank you, Swati, and thank you, IRCC, for giving me this opportunity to present. And today I will basically talk about chromatin. So basically, the topic is chromatin assembly, packaging DNA to encode extra information. So what we see here is a cartoon of chromatin, and I will explain what chromatin means and all the details in the coming slide. So as most of you know, DNA is a long polymer which contains the genetic code. It's a molecule that contains the genetic code, which means that all the information needed for a cell to function is supposed to be encoded in the DNA. For example, like when we eat food, how to digest the food is, the digestion of the food is done by a bunch of machines or molecular machines, and the DNA encodes this information on which molecular machine to make and what they will do. So DNA encodes that information. In another context, for example, if you have a wound, DNA encodes the information to make the material to heal the wound, as well as all the machinery to make all the machineries that would heal the wound. So all the information needed for the cell function is encoded in this very long molecule called DNA. And it's quite long. It is 2 meters long. The human DNA, if you just look from one end to the other end, combining all the DNA, it is 2 meters long, and it has to be packaged in a very tiny nucleus, which is about 10 micron size. So this, if you think of this ratio, it's like if you have a very tiny thin cable from here to CST, it's about 20-30 kilometers, and if you were to pack inside a tennis ball, that's kind of packaging that what is happening inside the cell to package DNA. So there is certainly a packaging problem, how to package such a long cable into a tiny space. So it's about nearly 10,000-fold packaging. There is also an information management problem because it's just not packaging. It's not just packed and kept there to store it. It has to be constantly used all the time, like as we speak or as we sit here or as we do any function, the cell is all the time doing function. So we have to read this information and produce proteins for the function. So how do you manage this information? We all know that we all start from a single cell and develop to a fully grown adult. So baby and an adult, right? So the single cell has DNA from our parents, and this DNA, when the cell divides, this information is copied multiple times, right? So all the cells in our body will have essentially the same DNA. However, we have very different cell types. For example, cells in our skin function very differently from cells in our brain or cells in our eyes. So even though the DNA is exactly the same, the cells show very different behavior. So how do they show very different behavior even though the code is essentially the same is an interesting question. And not only that, this, even if in a given cell, in a skin cell, or in a, so this DNA need to be read when you need it. You don't need any unwanted reading. So you have to prevent, for example, you want to digest things only when you eat food, or you want to heal the wound when only there is a wound, right? So you want to read it only when it's needed. So cell also use this packaging of DNA to code this information, that when, the temporal information, the time information. So cells need to package DNA such a way that some information is accessible and some are not, in a context dependent manner. So that is the job that cells have to do. So to do that, what we know from biology is that DNA in cells is not bare. They are covered by large number of proteins to form something called chromatin. So chromatin is basically DNA packaged with large number of proteins. So this cartoon, this DNA is packaged into some structure which is very complex and we don't fully understand. This is an EM picture of chromatin. So there is some way this DNA is packaged such that this function is possible. We now, in the last couple of decades research, we know that the fate or the function of a cell is not decided by the DNA sequence alone, but also by different states of chromatin. That is a summary of what I said so far. The fate or the decision is not just made by the sequence alone, but also by something which now we call as chromatin states, even though we still fully don't, we have not very clearly defined what chromatin states in the field itself. In people who understand the language of statistical thermodynamics, this is like a microstates, if one could imagine. So one could imagine a bunch of microstates, and one has to understand all these microstates to understand the state of chromatin. In textbook, we know that, what we know in textbooks is that DNA is packaged in many levels. There is a hierarchy of packaging. So this is a double helix DNA, and which is then further wrapped around this yellow ball-like proteins called histones to form a string of beads, and these beads are called nucleosomes. I would use this word nucleosome many times. Basically, DNA wrapped around some ball-like proteins are called nucleosomes, which are then further packaged into many levels, and we still fully don't understand. We know quite a lot about this level of packaging, but still we are trying to understand this higher order packaging of chromatin. And finally we have the chromosome. So when cells divide, if you look under a microscope, you would see mitotic chromosomes like this. So this is what the textbook picture of packaging, and in this packaging there is a lot of information encoded. So we all know that there is this information on the DNA sequence, but just not the DNA sequence information. On top of that, you can see those yellow dots. So DNA itself has some methylation marks, these red dots. And on top of this, there are these proteins that are assembled. You can see here that there is a lot of proteins here. So this DNA is inaccessible and you cannot read the DNA. Therefore, this information is inaccessible. So there is an extra layer of information because of the proteins, which tells that, oh, you cannot access this now. On the other hand, in these regions, there aren't many proteins. So those regions are accessible, which essentially tells this information that, okay, you go and read this now. So there is one layer of another layer is the protein layer. But on top of these proteins, there are some flags. So there are red flags here, there's a green flags here, and so on and so forth. So these flags are further chemical modifications, like they are called histone modifications, and there are other modifications on other proteins. So, but these modifications also carry some amount of information on when to read it, how to fold it, and so on and so forth. So the field is now trying to understand how all this information makes sense and how is it all being used to decide when to read, how to read, and also how to fold and package DNA. And beyond that, there is of course the flux of proteins. The green squares that I have drawn here are proteins that come from outside, in the sense that the food we eat essentially lead to a set of chemical reactions, and that would generate a bunch of proteins that all come and bind on the DNA, interact with the DNA. So in a flux of proteins, there is this molecule which has multiple layers of information, and how this all works is a very interesting puzzle which we only understand partly. So the question that we started with a few years ago is that to think about how are in this very first level of packaging, if we take the first level of packaging, we ask this question that are these nucleosomes organized randomly or is there a specific pattern of organization? So experimentally also we were thinking about this question, and what we knew from experiments is that they are not random patterns. There is a very specific organization of these nucleosomal proteins, and what is plotted here, this is from an experiment by Frank Pugh lab, which shows that the likelihood of finding nucleosome, the probability of finding nucleosome as a function of DNA along the DNA, if you plot, if you average over many genes, so this is a gene region, so if you average over many genes, you still get a pattern, which suggests that they are not random. If they were random, they would be kind of flat, but there is a particular pattern suggesting that they are not randomly organized. Then people also did single cell experiments looking at specific organization of these nucleosomes, and they found that when the gene is in the off state, predominantly you find a set of a particular kind of organization, and when the gene is on, there is a different kind of organization. So this fact that where the nucleosomes are positioned and how they are positioned also decides whether a particular gene should be switched on or switched off. So the nucleosome positioning does dictate some function or the fate of the gene or and eventually the cell itself. So our research basically asks the following questions. So we do theoretical studies using physical methods. So we use principles from physics to understand these processes. So we asked, how do different patterns of nucleosome organization emerge? So we know that experimentally these patterns, how do they emerge? So we made some models. So what a lot of physicists typically do is that they make different kinds of models and add components to the model and ask the question that what is the minimal model required to recreate the experimentally seen pattern. So this is what we did. So we had a model which has DNA along which proteins can bind, slide and dissociate with some rates, and these rates depend on various things including the sequence of the DNA, the ATP dependent processes that make them slide and dissociate, and so on and so forth. So there are many factors which I would be happy to discuss with you after the talk if you are interested in. Because of the lack of time, I would just quickly say that we had a bunch of rates modeling all the processes, and we solve these equations or we simulate them using kinetic Monte Carlo simulations and ask the question that what are the things that is required for obtaining the pattern. One interesting thing you see here is this violet color bar, or this is a protein which forms some kind of barriers at transcription start site, and it turns out that these are very crucial for obtaining patterns. So the work by Josna, who was a PhD student in the lab, she showed that if you want to get such patterns, what are the things needed? For example, we found that some particular activity by ATPases are absolutely necessary, and only then you would get this pattern. If you switch off those activity, if you don't have those activity in the model, you would not get this pattern, which is consistent with experiments from Frank Pugh lab. We then also asked how to get different organization which is seen in single cell level, and the student Humyo Kararin, who did work on this, and he figured out what are the minimal rules required to obtain the kind of organization that you would see in different cells and the distribution of nucleosome organization. So this shows different organization of nucleosomes, like nucleosome with all the three covered, two nucleosomes with different ways, and only one nucleosome and no nucleosome. So they all have some experimentally known frequencies, and we asked the question that what is the model that would obtain and what would be the parameter that would lead to this kind of model. So that's what one part of the work we did. Now the second part is, since nucleosome positioning is crucial, it is crucial for gene regulation, how do daughter cells achieve the same nucleosome positioning as that of the mother? When the cell divides, a skin cell divides, it has to stay a skin cell itself, right? So how do the same, during the cell division, DNA is replicated and copied, but how do they now copy this extra information on nucleosome positioning? So this is the question, during DNA replication, do daughter chromatin inherit nucleosome positioning or not is the question that we asked. So we asked, is it possible to transfer this information? Is there any theoretical model to think about this? And that's what we studied. So we made a model to study nucleosome positioning during DNA replication. So this was done by Tripti Bameda, who was a institute postdoc in IIT. And we constructed a model which is constrained by various known experimental data. There is a lot of data and details which I am not talking now because of the lack of time. But the simple essence of the model is that there is DNA being made into two parts, right? This is called replication fork. And as the DNA, as this moves, there are some nucleosomes here which has to be removed with some rate, which is 1 over tau p, and with some rate you assemble this. You also slide these nucleosomes, which are known in experimentally, and there is a time scale of sliding. And we have these parameters which are the minimal things needed to study this. So we have this 4 kinetic events, which is the movement of the fork, nucleosome deposition behind the fork, and sliding of nucleosomes behind the fork, and removal of nucleosome ahead of the fork. So these are the 4 activities, kinetic events, and we studied, simulated this problem with various parameters. Then we asked the question, so we know some of the parameters from experiments which are constrained, but those parameters which we do not know, we asked the question that what would be the parameter regime where you would get the inheritance that we want. So this is the parameter which is tau p, which is the pausing time, and tau s is another parameter. So we explored this parameter, and we, wherever this deviation is minimal, so which is here, where you would get this inheritance. So in other words, if the blue one is the nucleosome organization in the mother, and the red one is the nucleosome organization in the daughter, you would get the mother and daughter similar only when the tau p, which is the one of the time scale, is much larger than tau s. So which is in this part of the diagram. That's where the error would be, this deviation would be minimum. So this is what we find from our model. So basically our prediction is, if time scales of pausing is much higher than this time scale tau p, which is the time to reject this, is much higher than the time scale of sliding, how long the sliding will go on, then we can transfer this information, is what we predict. What is going on behind this I can explain, because I may not be able to explain in 5 minutes, but I'll be happy to explain this in detail if any of you would want to know about this. So that is another set of problems that we were doing. Then there is this second question, that what happens to higher order chromatin. So we know, we have been studying this level of organization. Then the question is, what happens, how do you go from here to here? So how do nucleosome, which is this, influence the 3-dimensional organization of chromatin, which was studied by Gaurav. So in the textbooks, there are some, already from some early experiments, there are some picture and there are some models. So the model says that the chromatin would organize like this, the nucleosomes would organize like this in a zigzag manner. So this is known as the 30 nanometer chromatin fiber, because this has a width of 30 nanometer. So this nucleosome has a width of 10 nanometer. So 2 nucleosomes 20 nanometer, and there is a 10 nanometer in between. So this is known as 30 nanometer chromatin. So textbook would tell about the current picture, or the picture a few years, still until a few years, was that chromatin falls into this 30 nanometer chromatin. But in the last decade or so, a bunch of experiments could do better than what they did earlier. So they all showed that if you do measurement closer to in vivo, you do not see this kind of regular organization of nucleosomes, and you do not see this 30 nanometer chromatin fiber. That's what all these experiments show. So for example, analysis of cryo-electron microscopy images does not support the existence of 30 nanometer chromatin fibers in mitotic chromosomes in situ. Human mitotic chromosome consists of predominantly irregularly folded nucleosome fibers without a 30 nanometer structure. So there's a bunch of papers, a series of papers, about 10 papers, which came in last 5-6 years, which all showed that in vivo they cannot see this kind of regular 30 nanometer organization at all. So people were puzzled, and we thought about making some models to understand why this is the case. So it turns out that earlier models only accounted for nucleosomes and DNA, but there are many other players in the problem. So we started making models accounting for other players, including histone modifications and some proteins that bind onto the DNA and bend DNA. So we basically made polymer models accounting for nucleosomes and DNA bending proteins. So this is the simulation that Gaurav did. And again, the details I would be happy to explain to you if you would want. And the result of our model is that as a function of the density of non-histone proteins, we would get irregular chromatin as they see in experiments. So experiments can quantify a particular way the irregularity of the chromatin. So we use those methods and ask the question, what should be the concentration of this non-histone proteins such that we would get this irregular chromatin that's seen in experiments. So that's what Gaurav did in this paper. And various interactions, so we explored systematically how various interactions decide the structure of chromatin. So there are many interactions, including electrostatic interactions and internucleosome interactions, because many of these are charged. The tails of histone molecules they are charged, and depending on the histone modifications, which I showed here, this acetylation, methylation, so they also alter the charge on chromatin. So accounting for this, what would be the chromatin organization is something which we examined, and that gave us some idea about how the nucleosome must be organizing given the various experimental constraints and information that we know. So we asked the question that what must be the critical concentration or density of non-histone proteins beyond which the so-called zigzag would disappear. Is that density biologically relevant? So this is some question that we asked specifically. So we had some way of measuring it by the probability of neighboring nucleosome to interact. For example, in this case, the neighboring nucleosomes would interact. The next neighbors, this is 1, 2, 3. So I and I plus second neighbors would interact, while in some other cases, I and I plus first neighbors would interact. So by quantifying this in a particular way, we can figure out what is the structure of chromatin, and you can see a transition from one state to the other state, which is from a regular state to an irregular state. So that's what basically this is. And it turns out that the non-histone protein density is about 30 to 50% of linger regions. If they are bound by non-histone proteins, that's sufficient to make it irregular. And this is consistent with various experiments that we know that about the number of, so the amount of non-histone proteins that bend DNA is roughly 2 to 1 for histones. So up to 50% is quite reasonable to have. So this is another set of work that we did on three-dimensional organization of chromatin. So to summarize the story, we asked the question, how do nucleosome positioning patterns emerge? And we showed that we can understand what is known from given some experimental data, our simulations can predict and learn something and predict some things. And so we generated kinetic models for nucleosome positioning during replication, and we try to understand what happens to nucleosome positioning and how this nucleosome information can be inherited during replication. And we also examined the three-dimensional organization of chromatin and the role of non-histone proteins. So that's basically the story of chromatin. I just show one more slide where we had completely different set of problems that we studied the organization of proteins themselves, which was done by another student called Srivastava Ranganathan, and we examined how protein organization can be understood from a physical perspective. Again, I would be happy to tell you the details. So to acknowledge the people who did the work that I talked today are mainly Josna, Hungyo and Gaurav and Tripti. So first three were PhD students and Tripti was a postdoc, and the last bit was by Srivastava Ranganathan. And I am thankful to all my collaborators, Dibyendu, Mandar, John Marco, Samir, all from IIT, except John Marco, all three are from IIT. And Ishitesh and Deepak their work I didn't talk about. They're also PhD students in the lab, and this is the current lab we have, and we are doing some future aspects of this work on three-dimensional organization of the chromatin as well as protein organization. So I'll be happy to take questions if you have, and thank you for listening.